Life relies on a cell's ability to maintain a distinct internal environment separate from its external surroundings. The cell membrane is the organelle responsible for creating a consistent internal environment for the cell by selecting the materials that can pass through the membrane and those that cannot. In this video, we will look at the structure of the cell membrane and how its structure allows it to perform its function as the gateway to the cell. We'll look at how the structure and properties of phospholipids allow them to spontaneously form bilayers. We will also examine the components of the membrane within the context of the fluid mosaic model. We'll focus on integral and peripheral proteins, glycoproteins, and glycolipids. These key concepts are some of the most important topics covered in the IB biology syllabus. The cell membrane is primarily composed of phospholipids. The phospholipid consists of a phosphate head and a fatty acid tail. The phosphate head is hydrophilic. It is polar and attracted to water. The fatty acid tail is hydrophobic. It is not polar and is repelled by water. We would call this molecule amphipathic, as it has both a hydrophilic and hydrophobic portion. For a more in-depth look at the phospholipid structure, you can check out the lipid section of the Key Concepts video called Carbohydrates and Lipids. Because of this, phospholipids don't just dissolve in water, as, for example, sugar might. In fact, they self-organize. When submerged in water, the tails are pushed together by the repulsion from the water, much like a droplet of oil tends to crowd together when combined with water. Because the phosphate heads are polar and hydrophilic, they will orient themselves towards the water while the tails orient themselves towards each other. This forms regular structures like lipid bilayers. These bilayers in 3 dimensions tend to form sheets, which we can see here. These sheets can then connect with themselves as spheres, like we see in the cell membrane. This image shows the membrane in 2 dimensions with a longitudinal cross-section. Because it will form these structures on their own just through the differences in attractions, we call them self-organizing molecules when they're submerged in water. This is essential to understanding how the plasma membrane functions. When small breaks occur, the membrane can spontaneously reform to repair itself. The fluidity of the membrane also allows for the movement of proteins and other molecules to move laterally through the membrane. This movement allows interactions between membrane proteins and other molecules. Because the membrane is fluid and self-organizing, it also has the remarkable ability for the membrane to bud and form structures like vesicles from membrane-bound organelles. The membrane can also absorb these vesicles in processes like endocytosis and exocytosis, which is explained in more depth in the higher-level key concept video on membrane structure. Now that we've discussed why the plasma membrane has fluidity, let's look at why the membrane is also referred to as a mosaic in the fluid mosaic model. The mosaic is an assembly of small pieces of material that come together to create a larger pattern, similar to how the plasma membrane is structured with various molecules that unite to form the barrier or surface of the cell. The plasma membrane is composed of components including phospholipids, proteins, and carbohydrates as a dynamic barrier. The phospholipid bilayer forms the fundamental structure of the membrane, and molecules that are either fully or partially embedded into the membrane, such as those seen here, and some of which span the entire membrane while others do not. These pieces are also fluid and can move laterally through the membrane and sometimes interact with each other. If a protein, glycoprotein, or glycolipid is embedded in the membrane, then they will have attractions that align with the membrane, which stabilizes their positions. So, for instance, a protein will have a hydrophilic region whose exterior amino acids are oriented towards the phosphate heads and watery interior and exterior of the cell, while the exterior amino acids that are in the portion of the protein embedded in the fatty acid tails are hydrophobic. The protein's interior may, for example, be filled with amino acids with a negative charge, which would allow a positively charged ion to pass through it. Because the interior of the lipid bilayer is hydrophobic, or nonpolar, large, polar, or ionic compounds can't pass through the membrane. For the cell to be able to move these molecules across the membrane, they must go through a protein. The cell can determine which substances and how much of them pass in or out of the cell by controlling the type or specificity of the protein, as well as the number of channels or protein pumps it has in the membrane. To those substances, the membrane is considered selectively permeable, meaning the cell determines which substances move in or out and at what rate that exchange occurs. However, the membrane is only selectively permeable to large charged or polar substances that require a protein to pass through. Because the membrane is constantly moving, it creates small gaps that molecules can pass through. Molecules like oxygen, carbon dioxide, and water can slip through the membrane through simple diffusion. Because the cell cannot control the movement of these substances, the membrane is not selectively permeable to these molecules. Protein channels or pumps are considered integral proteins, as they penetrate through the membrane and have portions that extend beyond the interior and exterior plasma membrane. An example of a protein channel would be an aquaporin that allows water to diffuse faster through the membrane by facilitated diffusion. An example of a pump would be the sodium-potassium pump. We discussed the function of these proteins in greater detail in the membrane transport key concept video. When comparing the structure of channel and protein pumps, we can see that pumps contain binding sites for the substances being transported. They can span the membrane multiple times to undergo conformational changes necessary for active transport. Pumps will use ATP, meaning they consume energy to function. Channels, on the other hand, will have a pore-like structure that forms a passageway for substances to diffuse through. They can have gates that open or close in response to stimuli. When comparing the function of protein pumps and protein channels, we can see that protein pumps are used in active transport, while protein channels are used in passive transport. This means that the pump protein must consume ATP because the pump is moving its target substance against the concentration gradient from a low concentration to a high concentration. The protein channel, on the other hand, does not need to consume ATP, as it allows the molecules to passively move from high concentration to low concentration in the process of facilitated diffusion. A membrane pump is slower as the protein needs to shift its shape in a conformational change in order to bring the materials across the membrane, which takes time. The channel, on the other hand, does not need to change shape unless a gate needs to open, and substances can simply pass through the pore, leading to a faster rate. Examples of protein pumps that are studied in the syllabus include the sodium-potassium pump, while examples of protein channels are aquaporins and ion channels seen in the nerve impulse. Integral proteins can have many additional functions. They can function as enzymes like lactase that are at the enzyme brush border of the intestines to break down lactose. They can also act as receptors, such as the neurotransmitter receptors in a postsynaptic neuron that initiates an action potential. Integral proteins can also act as antigens, or cell recognition in the immune response, such as the antigens present on red blood cells of different blood types. Peripheral proteins only extend beyond one side of the lipid bilayer and can project either into the interior or exterior of the cell. Peripheral proteins can also act as receptors for cell signaling, as well as membrane transport proteins that may shuttle compounds between integral proteins. HL can think of the effector proteins that are activated in G protein signaling. Glycoproteins and glycolipids are another class of membrane molecules that have carbohydrate chains attached to them that are composed of glucose and other sugars. Glycoproteins are integral proteins that have a carbohydrate chain attached. Both glycoproteins and glycolipids play an important role in cell recognition. Many antigens, molecules that elicit the immune response, are glycoproteins. They can also function as receptors for chemical signals like hormones, and also function to promote cell adhesion. HL might recall that glycoproteins create tight junctions to join cells to form tissues. In this video, we saw how phospholipids self-assemble to form the lipid bilayer. We also looked at the fluid mosaic model and saw that phospholipids and proteins are constantly in motion. We also discussed proteins in the membrane, which includes protein pumps and channels, which are integral proteins and have different structures and functions. We looked at other functions of integral proteins, which include enzymes, carriers, receptors, and antigens. We looked at peripheral proteins, whose structure does not span the membrane and can function as receptors, carriers, and structural support. Finally, we saw that glycoproteins and glycolipids have carbohydrate chains attached and can function as antigens and in cell adhesion.